Hi everyone, we are thrilled today to welcome to our Editor's Choice video program, all the way from Denmark and with affiliations to our Harvard Boston Dr. Rune Häckert Christensen, who is first author on this amazing work. And please tell us a little bit about yourself.
Thank you very much, Dr. Riggins. Well, as you mentioned, my name is Rune Christensen. I'm a medical doctor from the Danish Headache Center originally, where I'm also a PhD fellow. And this work we did, it was a joint venture, you could say, between Boston and Copenhagen. And I will tell you a little bit more about it later, but my story is that I've been doing headache research for a couple of years now, both in humans and now in animals as well.
That's strongly appreciated. Thank you for your amazing research. Could you please tell our audience a little bit about methods and study design? This article was done on animal models and very exciting findings.
Absolutely, absolutely. To begin with, I would actually like to give you just a short background of this study because it has sort of a funny story to it. It began in the clinic despite it being a preclinical study. And it actually began in Denmark where, of course, we have our patients with migraine, and as in any place, they commonly report certain triggers. So it might be that stress induces their migraine or alcohol or other things. And as many of you know, in Denmark, we have specialized in investigating what triggers migraine, what starts the migraine attack. Some of you might know CGRP or PACAP. But one very interesting molecule was levcromacalim, because when this is administered to patients with migraine, it's actually about 100% who develop a migraine attack. And we can actually see that it's the same for other headache disorders, that it can even induce cluster headache in individuals with cluster headache and migraine-like attacks in individuals with post-traumatic headache. However, the key question has been for us in Denmark, how does this exactly happen? And the reason for this is that from a very traditional point of view, levcromacalim actually has an inhibitory effect upon neurons. So it wasn't clear how exactly could it start a migraine attack, which one would think would require certain pain-sensitive cells to fire. And the background for this study was that we initiated a collaboration between Boston and Denmark. And the idea was, and what happened was that I went to Boston to conduct this actual study, specifically to the lab of Rami Burstein, who is specialized in advanced electrophysiological methods that can look at the specific cells, the nociceptors, that we think are responsible for migraine pain. So in this way, it was a very strong transatlantic collaboration between Masoud Ashina's lab and Rami Burstein's lab in Boston. So that was really the first piece of the puzzle, so to speak, to start addressing this question. The methods that we employ are some that I personally find very fascinating. And what we do is that we directly record from the trigeminal ganglion. So that is to say, outside of the brain in the peripheral nervous system. And then what you can do is that you can look at the actions potentials of the nociceptors there. So nociceptors are these pain-sensitive cells that we think are key to initiating the first signals of migraine pain. And our question was, what did levcromacalim do to these cells? And levcromacalim, to be specific, what it does on a molecular basis is that it opens specific channels, ATP-sensitive potassium channels, that are also very involved in vasodilation. So we wanted to know what did it exactly do to the pain system, essentially. And it's done in animals. It's done under anesthesia, but with the trigeminal vascular system intact. So it's actually a whole animal that you're looking at and investigating. And the methods we employed, we tried to align those very closely with our studies in Denmark, what we call the human model of migraine. So we gave a peripheral infusion for the same amount of time, 20 minutes, and then we recorded for several hours afterwards, how did these meningeal nociceptors respond to left chromacalim. So it was sort of taking a one-to-one from the patient to the animal to see what was going on. We like to call it going not from bedside to bed, but the other way, from bedside to the bench, to figure out what exactly was going on. So that is essentially the method. It's looking at these action potentials in the nociceptors. So a very, I like to think, objective and direct marker of trigeminal pain and headache.
That's amazing story. Thank you so much for this background. And I believe that you had the substance which was introduced into carotid right area.
Yes.
And what was the findings? It's so important to clinics. Please let
Yeah.
us know.
So we use a quite recently developed surgical method. It's developed by Agustin Melo Carrillo in the Bernstein lab, which is an intracarotid infusion within this setup, you could say. And the idea is that it might more directly target the headache-related or head-related pain mechanisms than, let's say, an intravenous infusion. That's at least the concept. And what we found was that we investigated 2 different doses, and we also gave a placebo, what is called vehicle in a preclinical context, to see how the nociceptors responded. And we found that while the vehicle did mostly nothing to the nociceptors, they activated quite robustly when we gave them the higher dose of left chromacalim, the dose which is also used in most other animal studies. In fact, it was the case that about 70% of the nociceptors, they became activated after this dose. So that is the main finding of this study, that left chromacalim and opening of these channels actually can activate the meningeal nociceptors, which from a purely pain perspective was quite surprising, but might give us some clues towards why it can induce migraine and other types of headache. And what I find interesting about this response in the meningeal nociceptors is that it actually aligned quite closely with what we see in the patients, in that the response wasn't immediate, it was delayed, occurring about 100 minutes or so as a median after we started the infusion. And it was also prolonged, so it wasn't just a brief effect, it was actually lasting for several hours and often past the end of the experiment, which was ongoing for 4 hours. So this sort of mimics what we see in patients with migraine. When they receive left chromacalim, it's not just a brief or transient headache, it actually mimics a complete migraine attack lasting for several hours, also with a delayed onset in many cases. So in that context, I think the findings actually align quite well with our clinical experience when we give or administer left chromacalim to patients with migraine. So that was quite interesting to see, I think.
It's
I might
extremely
elaborate.
interesting.
There's a few more details, I think, to the study which are worth noting. One is that we investigated this both in male and female animals, and we found that no matter the sex, the nociceptors actually activated at a similar rate, suggesting that, okay, this is a mechanism which seems to have a role for both sexes. And in the same way, we also electrophysiologically look at different types of pain fibers, what we call C fibers that are not myelinated, and then the thinly myelinated A delta fibers. So these essentially transmit different types of pain, one the slower and one the faster type of pain. And we found that both types were being activated by left chromacalim. Now, I might mention a little bit on why we find this a little bit surprising in the context of pain, because as I mentioned, left chromacalim has actually been found to have an inhibitory effect on neurons in general, at least, and perhaps even certain antinociceptive properties. So the big question is, of course, how does this make sense in the context of it activating these meningeal nociceptors? And what we think is that it could be that left chromacalim is activating the nociceptors in an indirect manner. That is, that it might not be acting directly upon the neurons, but instead through other mechanisms activating them. And a very prominent hypothesis in this regard is that it is through the vasculature. because we know that levetiracetam, it is a very potent vasodilator. It dilates the meningeal arteries, and it is thought that this vasodilation could actually contribute to activation of the trigeminal system. One hypothesis is that it's not only the mechanical dilation, but also potassium leaving the smooth muscle cells as levetiracetam acts upon the same channels to subsequently activate the perivascular afferents. So essentially a signal that goes from the vascular smooth muscle cells to the neurons. That is at least a working hypothesis. Another reason I find this study very interesting is that we have a standing hypothesis that it is possible that many migraine triggers might actually converge upon similar pathways to induce migraine, and that these pathways could involve the vasculature and specifically these potassium channels. Essentially, the idea is that certain neuropeptides, such as CGRP or PACAP, might in the end ultimately open the same channels and through the same mechanisms induce migraine attacks. So this could explain why we see an even higher induction rate with levetiracetam as compared to the neuropeptides. And from a therapeutic perspective, it's of course very interesting because it also holds the promise that we could perhaps target these channels to prevent and treat migraine attacks, and that it could have an even higher efficacy than, say, the antibodies targeting CGRP. So this is also why I think this study is a very interesting finding we have, and that it sows a certain hope for future treatments to come in the treatment of migraine and other headache disorders.
That's amazing. Thank you so much for this beautiful overview. I hope that our audience will take a very close look at not only at beautiful graphical abstract, but also in all details in this spectacular article in Journal of Cephalalgia, Editor's Choice. Thank you so much, and we hope to see more of your amazing work very soon. Thank
Thank you
you.
very much, Dr. Riggins.